Irimia from Lonnie, soon to be at USC. And he's going to tell us about neuroimaging of structural pathology and connectomics in traumatic brain injury. So, okay, please.
Okay, well, first of all, I'd like to thank you for this great opportunity to be here. I am a neuroimaging scientist in the Laboratory of Neuroimaging at the University of California, Los Angeles. And my primary research interest involves the combined use of multimodal structural neuroimaging, multimodal diffusion tensor imaging, and electrophysiological recordings to address important challenges facing the clinical community who are interested in improving outcome scores in traumatic brain injury. So in our group, we have adopted a tripartite approach to the study of TBI. As neuroimaging scientists, we are in very close collaboration with our clinical colleagues at the Ronald Reagan Medical Center at University of California, Los Angeles, where we acquire multimodal neuroimaging data sets from acute TBI patients. These are severe cases of TBI who are patients that are admitted to the neurointensive care unit at the hospital. And in addition to typical imaging modalities such as MRI and DTI, we're also interested in the use of magnetic resonance spectroscopy as well as functional imaging methods such as fMRI and electroencephalography, EEG. And we rely intensely on biomedical engineering techniques to analyze our imaging and electrophysiological data using novel techniques that, including information theory and network theory, nonlinear dynamics, and connectomics to explore changes that occur in the TBI brain throughout time. So to give you a brief overview of our research protocol, I'd like to show you a set of longitudinal MR images that are acquired from several patients in our hospital. As you can see, these are multimodal data sets, and they are also longitudinal data sets. We acquire data at 2 time points. One of them is acute, approximately less than 72 hours after admission to the medical center, and chronically at about 6 months after injury, so that we can study more with improved accuracy the process of healing and repair in the brains of these subjects. The first and the most important challenge that we have faced in dealing with these data sets and in analyzing these data involves the combination of modalities to perform segmentation of tissues in traumatic brain injury. And as you can see, we are using several types of imaging techniques, which I'm sure you're familiar with. The MPRAGE T1 and also the FLAIR GRE T2 and SWI modalities, which give us the opportunity to image CSF perfused edema in the case of FLAIR, as well as hemorrhage in the brain in the case of GRE and SWI. The challenge that we're facing in the case of TBI segmentation as opposed to segmentation of adult brains from which are normal or which have less severe pathologies such as Parkinson's or Alzheimer's is that we have to account for a large extent of structural complexity in dealing with the size and position and type of lesion that are present in the brains of these subjects. And as you can see, the lesion profiles can be very heterogeneous from patient to patient, and it is often difficult to separate the tissue types and to characterize each voxel and to assign it a certain pathology type. And what we have done is that we have acquired a semi-automatic segmentation approach in collaboration with our colleagues at the University of Utah, led by Dr. Guido Gehrig here in the audience. And what we have performed is a quantification of longitudinal changes based on these semi-automatic methods of segmentation. And as you can see, we're able to segment the cerebrospinal fluid swelling and blood that occurs in the acute case in these TBI patients, and to localize and characterize the type of lesion in each case. And then at 6 months after injury, we're able to characterize the necrosis and the bleeds that are still existent in the brain, as well as the changes that have occurred between the two time points. We are, aside from these challenges, we're also interested in quantifying morphometric alterations to the brain. And in this type of visualization, what we have done is to overlay the acute, the structure of the brain at the acute time point, which is depicted in red, to the structure of the brain at the chronic time point, 6 months after injury, shown in green. And as you can see, there are quite a bit of differences. Here, there is a lot of red, which is, in this case, due to an injury, a primary injury in this patient. But also, there is a lot of, there's a midline shift, which can be seen by this area where red overlays the green. And then after 6 months, we can see a dramatic change in the position and structural profile of the ventricular system. And there have been a number of studies that have connected the structure of the ventricular system to outcome scores in traumatic brain injury. And from there, we would like to also explore how diffusion imaging can be integrated into this type of application in order to study changes to the white matter structure of the brain. And as you well know, there is a lot of heterogeneity in TBI. Here you can see there are 3 cases of acute TBI with different lesion loads and different types of pathology, with red representing hemorrhaging brain and green representing edemic non-hemorrhaging brain. And what we have done is we have undertaken the task of segmenting the gyral and sulcal structures of the brain to create a parcellation scheme that can allow us to integrate our DTI processing techniques with this type of application. And we have chosen a different color scheme for each lobe of the brain, and within that color scheme, we have colored each gyrus and sulcus using a different color. So, using these methods, we have gone on to map the connectome in traumatic brain injury. So, starting with automatic segmentation of the cortex and subcortical structures, and the DTI tractography that we are able to perform, we have performed this automatic parcellation of the gyri and sulci in the brain, and then determined the connectivity matrix of the cortex. So this connectivity matrix is an n-by-n matrix which contains entries for each gyrus and sulcus, and then there is an entry in the matrix if there is a connection between the region represented, say here, and the region represented, say here. So using these type of techniques, we have proposed a type of visualization which is called a connectogram. And this is a comprehensive visualization of the TBI connectome at a scale that has been previously unavailable. And before I say more about this, I'd like to explain what connectograms are. So first of all, the connectome is the complete ensemble of white matter connections in the brain, and the study of the connectome is called connectomics. So one of our contributions to the field is the introduction of this visualization technique called the connectogram, where you can see the left and right hemispheres of the brain represented. The subcortical structures are represented here. Then we have, within each lobe, we have each region of the cortex represented either as a sulcus or a gyrus, with a color on the outside corresponding to the color of the structure on the cortex. And so on, we go for insular cortex, limbic cortex, temporal, parietal, and occipital cortex. And each region of the cortex, whether a gyrus or a sulcus, is represented by a different type, a different color within its own color scheme. And the links that connect any two regions are connections between either a gyrus or a sulcus, between any of the regions involved, or perhaps connections between the subcortical structures and cortex. And the interesting thing that can be done with these types of visualizations is basically encoding, performing a systematic encoding of the white matter and cortical properties of the brain. And this is the legend for the connectogram inner circles. We have a parcellation code representing each parcellated gyrus or sulcus. And then on the inside of the connectogram are color encodings of gray matter volume, parcellation area, mean gray matter thickness, mean curvature, and degree of connectivity of that region. So the outside of the connectogram shows the structural information associated with this patient. And then we have encoded also for each link between regions, we have encoded FA in the color so that the lowest tercile of FA is colored in blue, the middle tercile of medium FA is displayed in green, and links that have high FA are shown in red. So what we have done, first of all, before analyzing before applying this methodology to TBI, we have applied it to a set of 110 healthy human adults of age 25 to 36. This was published in Neuroimage. And what this represents here is a systematic representation of the human connectome, of the average human connectome for this normal healthy adult population at a resolution and degree of systematization which has previously been unavailable in the scientific literature. And most interestingly, we have quantified how TBI modulates atrophy in the context of the human connectome. And these are three connectograms from three different subjects showing connections involved in primary TBI. So all the regions, all the links displayed in each connectogram denote regions that were involved in the primary injury of the patient. And then 6 months after injury, we have repeated the process, created connectograms, and performed the analysis for all these patients. And we have found various degrees of atrophy in the brain of each patient along the white matter fibers connecting various regions. And as you can see, although there is some agreement, there is quite a bit of heterogeneity in the extent to which the brain of each patient is atrophied, and there is no clear correspondence between the location of primary lesioning along white matter fibers in the acute time point compared to the chronic time point. So this highlights that we really need a systematic connectomic approach in order to evaluate how how injuries to the white matter either heal or atrophy after injury. And I would like to say a little bit about how we are taking the next step to integrate this type of structural neuroimaging involving MR and diffusion tensor neuroimaging with electrophysiological data, and specifically with inverse localization of EEG data acquired from acute TBI patients that were in the NICU under continuous EEG monitoring. And what we have done is we have combined all the MR modalities that we've had access to with CT imaging and created a high-resolution finite element models with as many as 400,000 icosahedral elements and as many as 25 tissue types included in the model in order to accurately account for every tissue type, including pathology, including different types of pathology. And based on this type of models, we have gone on to generate forward and inverse models of EEG in traumatic brain injury. So these are, with 25 tissue types, by the way, this is the most accurate finite element model that we are aware of being available in the scientific literature so far. And based on these recordings, we have looked at the sensor space EEG waveforms that are recorded by the EEG sensors in the NICU from these patients. We have isolated epileptiform activity, such as this epileptiform spike here. Here is the display in sensor space, and this arrow represents the localization of this negative deflection in source space, namely on the cortical surface. Why is this important? It's important because about 30% of TBI survivors are prone to post-traumatic epilepsy, and thus far it has been extremely difficult to perform neurosurgery for elimination, for resection of these epileptic foci due to the inability to accurately localize them in the cortex. However, with the advent of this type of technique, which is extremely accurate and extremely advanced from the standpoint of the number of tissues that are represented, we are able to push this to the next level and perform accurate localization of epileptiform activity with a high degree of accuracy. And I encourage you to look at this paper that has just been posted online in Clinical Neurophysiology for more details on this. So finally, just to show you as a cameo of what we have done and just as an example of our work, I'd like to show you this visualization about a very famous case in the history of neurology, which I'm sure most of you are familiar with. It's the case of Phineas Gage, who in the 1840s, had a rod pierced his head when he was working on the railway in Vermont, and I'm sure most of you are familiar with the details of the story, but the idea is that this individual had a rod, tamping iron, piercing through his head and destroying part of his brain, and miraculously he survived and recovered to live another approximately 20 years. What we have done here is we have created a sophisticated environment to fit the brains of 110 normal human adults with that had his age and demographic characteristics. And we have created basically a simulation of the white matter fibers that would have been destroyed by the tamping iron as it went through his head. Oops. And I have a video, which let's see if I can get it to run. Maybe I need a little help with that. Okay, so this is just a rendering of the case, and it shows our modeling, including the CT skull of Phineas Gage, which we recovered from Harvard Medical School with thanks from Dr. Ron Kikinis. The skull of the patient is located at the Harvard Medical Museum, and this is a representation of the white matter fibers that were destroyed and some of the ones that were still left after injury to the patient. So this article is one that I encourage you to look more, further into. It's been featured in the press as well as a featured story in Discover magazine, I think last year. It's a very interesting attempt to reconstruct some interesting features about this very important case in the history of neurology. And well, I guess I only had a few other slides. I just wanted to encourage you to take a look at our papers that on this set of topics, and I'd like to thank all our collaborators, including the group of Guido Gehrig. Okay, so here is the connectogram of Phineas Gage and showing the connections that were destroyed in his brain. And these are some of the papers that describe the work that I presented today, and I encourage you to take a look at them. We've been featured on the Journal of Neurotrauma on a special issue on biomedical imaging, biomedical engineering for TBI. The connectogram has been featured on the cover of Neuroimage, and we've had some of our DTI work in brain imaging and behavior. I encourage you to look at all these very interesting and exciting efforts we've looked at. I'd like to thank Dr. Veneer and all the organizers of this exciting workshop, our group at UCLA, our group at the University of Utah led by Dr. Gehrig, who spoke earlier, our collaborators at Kitware and UNC Chapel Hill, Harvard Medical School, Dr. Ron Kikinis, UC Irvine, Boston University, Dynamic Consortium, which sponsors our work through these grants, and the Human Connectome projects, which we are actively involved in. So I'd like to thank you very much for your attention.